DNA to protein. In eukaryotic cells, transcription occurs in the nucleus where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make messenger RNA strand with the help of the enzyme called DNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the genes function as a recognition site for RNA polymerase to bind. When DNA replicates, its strands are separated by the enzyme helicase. Then, during elongation, the RNA polymerase slides along the template DNA strand as a complementary bases pair up. The RNA polymerase links the nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminated portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The lagging strand of the DNA is a little more complicated. The strand is going in the opposite direction, which causes the DNA polymerase III to synthesize fragments called Okazaki fragments. These fragments are then put together with an enzyme called ligase. The strand of messenger RNA that is made during transcription includes regions called exons that code for protein and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed and modified, such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing, and is performed by a complex made up of proteins and RNA called the spliceosome. This complex removes an intron segment and joins the adjacent exon to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. Messenger RNA to protein. Messenger DNA strand is translated into a protein. The nitrogenous bases are grouped into 3-letter codes called codons. These codons code for specific amino acids. There are 4 special codons that code for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transferred RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called E, P, and A sites. Individual amino acids are brought to the messenger RNA strand by the tRNA complementary base pairing. As the complex slides down the codon to the right, the tRNA exits the E site and the A site is open to the next transfer molecule. As this continues, the polypeptide is being made as each tRNA brings in a new amino acid. Once the stop codon is reached, the polypeptide is released from the transferred RNA in the P site. The entire complex dissociates. The polypeptide that was created still needs modification. Modification is done in the endoplasmic reticulum and is modified as it passes through the Golgi apparatus and then is secreted using a vesicle that passes through the membrane and is taken wherever it needs to go.